Excellent. So we have about a minute or so left before we begin. But before we get into dissection, I know you're probably all looking at the photo of the brain there, and you just hear a disembodied voice going on there. So let me just go on to camera here. So hi everybody, my name is Alejandro. So I'm one of the STEM educators at Liberty Science Center. So as you all are maybe doing, I am working from home as well. So I'm here live streaming here in my living room, bringing you some really awesome educational content, all right? So before we begin, I know from the past few live streams that we've done, so Miss Caroline and I have been doing the live from surgery series every Tuesday at 1 p.m. And I know that we had a couple of dissections going on during our time live streaming through the LSC Facebook page, such as a cow's eye. I know Miss Caroline did a sheep heart. Today we're going to do a sheep brain dissection. And I noticed that a lot of you, whether you're homeschooling or you're from a school, I know that a lot of you probably are taking biology class at this time of spring that usually might have done a type of dissection. I know a lot of anatomy and physiology students out there will probably be doing some of these dissections at school as well. So I'll give you some time. So I'll be looking at my second screen here to look at some more comments and some facts because I asked for some facts. But if you would like some time, and if you are planning to write down notes during our presentation, feel free to do so. If you would like to take photos of you interacting with us, sending us questions and comments during our live stream, definitely post it on your social media, and always hashtag Liberty Science Center, all in one word, all lowercase, no spaces. We definitely would love to see how you're all interacting with us. But if you need some quick time to get a piece of paper and a pencil to take down some notes, feel free to do so. I'll do a little quick shout outs here and there, and then we'll begin. All right, so I know I saw a lot of people from Connecticut, a lot of New Yorkers, a lot of people from Pennsylvania. I know that I saw North Carolina, South Carolina, even Arizona. So thank you all for taking the time out of your day, working from home, learning from home, to come join us and do a brain dissection. So I know a lot of you were dropping me some comments here and there. So please drop off some more comments, some fun facts about the brain while I go through them. And I know some of you started dropping some of those bits of knowledge in the comment stream. So I see that the brain is squishy, so that is very true. The brain is very, very squishy. So we'll talk about that in a little bit. A lot of you mentioned that there are different lobes to the brain that pretty much oversees different types of functions. We will definitely look into that as well. I see someone did an owl pellet. So Marissa's mentioned that they did an owl pellet dissection. Beautiful. Those are pretty fun to do. We got a lot of third graders out there. We got someone who's 8 years old. Welcome, welcome. Beautiful. Second grade, Pennsylvania, Virginia. I absolutely love it. So just to give you an idea of what we have been doing during our time here on Facebook Universe out here, we've been doing a lot of live streams. So the Liberty Science Center has been hosting a lot of live streams from Monday through Friday on different topics, different themes, all at different times. So the Live from Surgery series is a live stream that happens on Tuesdays at 1. So if a lot of you have been coming back and checking out some of those streams, welcome back. If this is your first time, welcome, welcome. Hopefully I do a great job of, hooking you in and coming to check out some more of those programs. But it is all pretty much inspired by our Live from Surgery program that we actually do at the Liberty Science Center. So during the school year, we invite different school groups to come to the Science Center, and they actually get to see surgery done in real time. So they get to watch surgery and as well as interact with the surgical team as they're performing those procedures. And so as we are watching, the surgical team will be answering questions. We have a nice new format too, where we have the surgeons come in, interact with the students, and talk to them all about their career and some of their knowledge of their area of expertise, and then they get to watch some surgery. So what we try to achieve here during our live streams is not only get to understand human anatomy and what is the function and what happens when something needs to be solved, if there's a problem that may arise in said anatomy, but we also try to achieve to inspire and prepare the next generation of medical professionals, whether they're doctors, anesthesiologists, nurses, even outside the OR. We may look for scientists, we may look for engineers. So we try to aspire to provide that spark of interest when we do our programming in Live from Surgery or any of the programs that we offer at Liberty Science Center. Of course, a lot of that goes into putting a lot of hard work. So right now you may see me on screen, but there is a whole ton of people behind the scenes that come together to perform the operations that are needed to provide the content that you are seeing right now. So as a nonprofit organization, LSC relies upon donations of all amounts to present this and other programs. So I do want to give a shout out, because I know I've been saying saying hello and shouting out to people who have been saying hello. I definitely want to give a very special shout out to all of our members and some of our huge supporters, because without them, we wouldn't be able to do these live streams for you all today and all the other days. So definitely thank you all for doing so. And if you are interested in supporting us, definitely visit lsc.org/donate at any time. I know a couple of you mentioned, will this live stream be available afterwards? Absolutely. So if you have students that couldn't log on, they can definitely check it out. Just go to lsc.org to check out the video. All right, so let us begin. All right, I've been talking for too long and I'm not showing off anything interesting. All right, so what we are doing today is a sheep brain dissection. All right, I'm going to show you on screen. It is just the brain itself. I don't have the whole animal here. All right, so the way that we perform dissections is that we only have the small bits of specimens for the dissections. We don't own a farm. We don't go out and look for these animals and, dissect them for research or anything like that. We actually work with a company that supplies us these dissection specimens. So all these animals that we know have been treated humanely. A good portion that we know from the company that we use are middle person. These animals have died of old age, and so we are able to have the privilege to do these dissections for the basis of learning and gaining that bits of knowledge. So on top of that, I just want to give a nice little shout out to the animal that was, able to give us this opportunity to make us have this dissection available for you all. All right, so let's start with our dissection. Okay, so here we go. We have the brain specimen right here. All right, so this is a sheep's brain. All right, and before I forget, I do have my colleagues, Ms. Caroline, if you remember from all the other live streams, and Ms. Rosa. They'll be searching through the comments thread to answer any questions you may have, so feel free to give me some of those questions as well. I'll do my best to look over and answer those questions down on the comment stream, but I'm going to make sure that you get the most out of this dissection. All right, so here we have, as I mentioned before, is our sheep brain. All right, so this is the external anatomy of this organ. All right, so as I mentioned before, we definitely use dissections to learn more about anatomy. So if you remember from my cow eye dissection and the very few rounds of dissections that we were doing, this is all pertaining to comparative anatomy. So comparative anatomy, pretty self-explanatory, is we are utilizing different types of species to understand what anatomical structures are similar throughout all living organisms, and how do they differ depending on the organism that we are examining. So, with that being said, a lot that we are going to see with our dissection here, there are a lot of similar structures when it comes to the human brain. There are some differences here and there as we go through our dissection, okay? But I will point those out as we go along, okay? So, I know a lot of you were mentioning that there are different lobes to the brain that cause different functions. I know a lot of you were mentioning that it connects to the rest of the nervous system. The brain is certainly squishy, so it is a very pliable organ. All right, so this is an adult sheep brain. Oh, we got someone from Hawaii. Welcome, welcome. And so the average weight of a sheep brain is actually about less than a pound, I would say. It's not that large at all. I think, if I'm not mistaken, it's about 140 grams. Okay, so even smaller than that. But this is the same idea in terms of how the human brain looks like. So if you're wondering how heavy, what's the mass of a human brain, an adult-sized human brain is about 3 pounds. All right, so as you can see, it is, we're going to look at all the external anatomy. So I'm just giving you a nice 360 view, the top and the bottom. So just before we even start the dissection, I just want to give you a little bit of science facts in terms of positioning. All right, so we have front, back, we got this part right here, and then the bottom part right here. So when we dissect specimens, we like to use some scientific terms. All right, so we have, when we talk about this area right here, the front of our organ, we call that the anterior portion of the organ. When we talk about this back portion here, we talk about posterior. All right, so anterior, all it means is that it's the front part of that specimen. Posterior means it's the back part, okay? When we talk about this area right here that we're looking at right now, it's called the dorsal section of the specimen. So you probably heard of dorsal fins. All right, these are, you know, with whales, dolphins, sharks, they have that fin that sticks out on their back. That's the dorsal side. So for us, our dorsal side is our back. When what you're seeing right now on camera with my face and my chest and my abdomen, my torso, that's the ventral side. So when we look underneath, that is the ventral side of the brain. Okay, so let's look at some of that external anatomy. Okay, so someone mentioned that it's divided into 2 pieces. You're absolutely correct. So, as you notice, and actually, you know what? Let me zoom in much closely so we can get a nice view. All right, so when we look at the brain, all right, we have, it's divided into what we call 2 hemispheres. We have the left hemisphere, which is this side right here. We have the right hemisphere right here. Okay, and then you notice that there is a long line that kind of divides the 2. We call that the medial longitudinal fissure. So it is actually a very deep groove that separates both our left and right hemispheres. So you notice that we have a line, a boundary right here. We call that the transverse fissure. So this transverse, all it means is that it goes across. So we have a division that goes across, and it actually divides this part of the structure here compared to the rest of the structure. So when we talk about the hemisphere, the left and right hemisphere, that longitudinal fissure, we're talking about one big region called the cerebrum. All right, so you notice that the cerebrum is the largest part of the brain, largest section. You see that there's a lot of wrinkles to it. All right, there's a lot of major functions that go on there. So when we go on with our dissection, you'll see some of those parts. So this is the area where we get our conscious experience. So we understand and process our perceptions, our emotions, thoughts, and planning. Okay, So that is the cerebrum. This part right here is the cerebellum. It means little brain. And the cerebellum, one of its main functions is to ensure that it can control movement, balance, and muscle coordination. So all that happens in this region right here. And what we see sticking out in the posterior is the spinal cord. So some of you mentioned that the brain is connected to the spinal cord. So this is the beginnings of that spinal cord. So if you're not sure what the spinal cord is, the spinal cord, think of it as a giant cable, a very thick cable. And it's a bunch of nerve fibers inside the spine that connects to the brain to different sensory and motor nerves throughout the rest of the body. Okay? So that's the spinal cord. All right, so we mentioned lobes. Okay, so I'm going to give you some idea of where the lobes are, because some of the things when it comes to brain dissections, you'll notice that a lot of the structures look very similar to one another. All right, so I will do my best to kind of differentiate for you all. Okay, so let's go back to our brain. Now, the first lobe we will see is the frontal lobe. So the frontal lobe is this area right here at the very top in the anterior. So if I were to talk about our frontal lobe, if I were to touch my forehead, that's where the frontal lobe is located. Okay, so that's the frontal lobe. Okay, so the frontal lobe controls all cognitive processes. So when we talk about cognitive processes, we're talking about executive decisions. So this is where the center for planning and understanding your morals of what's a good idea, what's a bad idea, you know, planning before you start making a move, all right? That is where all of that process happens, okay? Just to give you an idea, the frontal lobe for the sheep is very small compared to humans. Humans have a very large frontal lobe, okay? That doesn't necessarily mean just because they have a small frontal lobe doesn't make the sheep unintelligent, all right? The larger the frontal lobe found in an organism, it is a small indicator that they have the ability to plan, to make those executive decisions, all right, to plan those things out. In fact, for humans, it accounts for 25% of the brain volume in our heads, all right? So that is the frontal lobe. The next thing we're going to look at is the parietal lobe. So the parietal lobe is actually right above, right up here. All right, so you can see there's the two frontal lobes. There's the parietal lobe. All right, so the parietal lobe integrates all the information that we get from our senses. It also helps us with spatial recognition. So if we were to talk about the parietal lobes for us, it's pretty much right at top. All right, so that helps us with depth perception, understanding our environment, just to get a nice view and spatial awareness. Okay? The next part, which I'll go towards the bottom here, is our temporal lobe. So we have one here, and we have one here. All right, so the temporal lobe helps with auditory perception and long-term memory. So what that means is, for us, our temporal lobe is located on the sides of our head pretty much. A major portion of our temporal lobe are on the side of the heads. So if you look at the side of your heads, all right, and you point your finger, the next structure that's actually very close to it in proximity is your ear. So your temporal lobe handles all that auditory information and helps processes it. It also helps with long-term memory. So it helps you remember your parents' birthdays, for as an example. So that's something that, you know, that's stored deep in there, okay? So let's move forward, okay? So I mentioned all the deep grooves, all right? So when we talk about the grooves, we talk about these little ridges up here. So I have it on my probe here. Those big hills, we call it hills and valleys, just for our intents and purposes. We have those little black lines. those are our valleys, and then we have those little hills, okay? The little hills is called the gyrus. So those are the bulges that we see on the brain. Those little valleys that you see, that's kind of stained with preservative, we call that the sulcus, which are all the dips into the brain. So when we make the incision, you'll see all those gyri and sulci. All right, so let's move quickly over at the bottom because there are some important structures that we need to see there. All right, so real quick question I'm going to look over. I see Jaden has a question. Does sheep have spines? Excellent, excellent question. So, very honest question. So, sheep is under a large group. So, if we were to classify all these different animals in a giant class system, all right, sheep, bony fish, humans, cows, goats, so on and so forth, we all land under a huge class called chordata. So chordata is a special scientific term, I believe it's Latin in origin, that means spinal cord. So these are animals that have a spinal cord, and so with that being said, the spinal cord is super important, so it needs protection. So they have that spine to protect that, that long anatomical structure. So very good question. All right, so let's go back to our brain, okay? There are some nerves that we can point out. All right, so what we see here is two nerves right out the gate. So it's not very pronounced here. We can see it right here. So actually, can I lift it up a little bit? So if you notice, I'm lifting it up a little bit. So this right here is called the olfactory bulb. All right, so there's one here, so you can see it right there. And there's another one right here. The olfactory bulb is involved in detection and determining different smells. All right, so humans also have an olfactory bulb. So the olfactory bulb is pretty much lined up right where our nose is, okay? So the olfactory bulb, you notice it's very thick here in structure for the sheep. When it comes to our olfactory bulb, it's not as pronounced, it's not as big as a sheep. And that's because smell is super important for the sheep. In fact, any animal that has a very long snout, they rely a lot on smell. So their olfactory bulb, that nerve, is very large when we examine their brains. On top of that, the sheep also relies on smell for something called imprinting. So imprinting is when you have a small lamb, they would smell for their mother. And once they pick up that scent and understand that that scent is their mother, then they will stay with their mother as they grow up. So that's called imprinting. So that actually happens immediately after they are born. All right. Next group of nerves. All right. So we have this area right here. Unfortunately, for this area right here, depending on how we receive our specimens, sometimes they're not all in one piece. So I'm going to do my best to point it out for you. But this is actually 3 separate structures in this area right here. All right, so this little portion right here is the optic nerve. So you'll see one that should be here. That's also the optic nerve. So if you remember from our cow eye dissection, if you saw that dissection, the optic nerve helps us transmit all that visual information from the eye all the way to the brain. You notice that there is a crisscross that happens here. So we have this optic nerve kind of crisscrossing over here. If we have this nerve here, it would crisscross over here. That point of crossing is called the optic chiasm. All right, so that helps us with binocular vision. All right, you notice that this optic chiasm is very pronounced in the sheep compared to humans. All right, the reason for that is sheep are considered prey animals. So if you ever notice an animal that has their eyes on their sides of their head instead of on the front like humans and tigers and lions and so on and so forth, we tend to be predatory animals, all right? So we have our eyes in the front, so that gives us a nice view of binocular vision of in front of us. For prey animals that have their eyes on the sides, that actually kind of extends their view so they can have a little bit of peripheral vision from behind them. So that gives them, for safety purposes, in order to know that they are safe in their environment. All right, so because of that, it's going to be a little bit more pronounced. And then the back portion is the optic tract. So that's just the nerves that continue all the way to the brain. All right, and I forgot to mention the other lobe now that I'm talking about the occipital. All right, so now we have the frontal lobe, the parietal lobe, the temporal lobe, and now that I mentioned vision, this back portion right here is the occipital lobe. So these optic tracts, if we were to isolate these nerves, it'll actually go to this back portion right here. So the occipital lobe handles all the visual information and helps interpret it for us. So for us, our occipital lobe is actually on the back of our head, so right back here. So if you were to place your hand on the back, that's where your occipital lobe will be located. Okay, so let's move forward. Now, you notice this area right here, so from this area about here and all the way down here is called the brainstem. So the brainstem is a giant route that transfers information, I should say, all right, between the spinal cord and the rest of the brain and then back again. So it's kind of like that highway of information. So the brainstem actually divides into more structures. So we have this portion right here. So you see this small portion right here. So you have a line right here and a division right here. So this portion right here is called the pons. So that's one of the connections between the rest of the brain to the cerebellum and the medulla. So that's the next structure we'll talk about. So that is pretty much the highway system for the cerebellum in the back, for the rest of the brain, for the spinal cord, so on and so forth. All right, the pons is also responsible for regulation. So it regulates sleep, breathing, it helps with swallowing, it helps us control our bladder so we can go to the bathroom and pee. It helps us with auditory information, taste, eye movement, facial expressions. There's a lot that goes on there. So that's just a small list of what happens there. When we look at the medulla, you might know it as the medulla oblongata. All right? So, when we look at the medulla, all right, that connects to the spinal cord. It's also very important what we call autonomic functions. So autonomic functions are things that happen in the body that we really don't need to think in order for those functions to happen. So that's a lot when it comes to our heart rate. Our heart rate is an autonomic function, so it just keeps beating. We don't have to think for it to beat, it just beats on its own. All right? It also helps with swallowing, and it also helps with respiration as well, or breathing. All right. Now we kind of covered all of the information there. Okay, so just to give you an idea, if you are writing notes, here are the external features right here. Okay, so we have the lobes. All right, we have the hemispheres. We have the cerebrum, which is this area right here. We have the cerebellum, which is divided by the fissure. We have the longitudinal fissure. We have the spinal cord. All right, the gyri are the little ridges, the little bulges. The sulci or sulcus are the little grooves between each gyri. Okay? And real quickly, on our ventral anatomy, we have the olfactory bulbs, the optic nerve, the optic chiasm, the optic tract. All right, we have the temporal lobes on the sides here with the frontal. So now we have the right hemisphere here, the left hemisphere here. We have the medulla right here. We have the pons, and then that region's the brainstem, and then we have the spinal cord. All right, so now let's get to the actual look of what's inside the brain. Okay? So, when you go to school, especially in high school, or if you go into biology in college, you're going to be doing a lot of dissections, especially if you go to med school or veterinary school. There's a lot of dissections that occur in order to understand the anatomy. So one of the big things that we learn in class before we do any dissection is that because we have the privilege of checking the anatomy out, studying the anatomy with something tangible like a dissection specimen, we have to give it the utmost respect. All right, so that means when we perform our dissections, we have to be careful, not only just so we can look at the structures as a whole, but not just go in there and start cutting everything up because we can. All right, we have to give all of our dissection specimens the dignity and the respect because at one point, these were living things. All right, so if you think I'm going a little too slow with the cutting, that's because I'm trying to be careful to not only make sure that the structures aren't damaged so you can see them, but I want to make sure that I'm giving it as much respect as possible by not just going in, just slicing it open. All right, so here we go. So I'm going to take my scalpel and I'm going to cut along the longitudinal fissure. All right, so it might come off camera a little bit. I just got to situate it so I can cut through. So I'm going to use the longitudinal fissure to perform the incision, to perform that cut. So as some of you mentioned, it is pretty soft. It's pretty easy to cut through. So it's one of the advantages of doing a brain dissection. It's very easy to dissect through. And a lot of the organs, a lot of the structures inside usually remain intact. So there we have the cut open brain. All right, so you notice that there's a lot of different structures. Okay, here we go. So we actually have different regions. All right, so let me bring that up real quick for you. Okay, so what you see here, this portion right up here, all the way here, is called the forebrain. So that's the region that has the cerebrum. So that's, as you can see, is a very large portion of the brain. Okay, the next thing we have is the midbrain. So I'm going to do my best to kind of like isolate it here. So the midbrain is right here. So the midbrain is that region that connects the forebrain to what we call the hindbrain. So the hindbrain is this back portion of the brain. All right, so we got forebrain, midbrain, hindbrain. Okay, so the hindbrain, as you can see, houses the medulla, the pons, and the cerebellum. The forebrain houses all the other major structures. All right, so let's go back to our dissection. Okay, so there are a lot of different structures that we see here. Okay, and same thing. One of the, for someone who is learning this for the first time, a lot of the structures look the very same. So I'm going to do my best to identify all the structures that we can, and hopefully you can differentiate them as I go through them. Okay? So, let me just cut this off right here. There are some connective tissue there. So I'm going to, let's see which side would be best. So let's go with this side first, because we have a nice view of the ventricles. All right, so this is actually a beautiful view of the ventricles. So, what you see here, you see this cavity right here? This is what we call the lateral ventricle. So the ventricle is actually on the other side as well. So it's not as open here. So actually, I can open it a little bit right there. So that is ventricle. So there are four ventricles in the brain. So we have the lateral ventricle. We also have one here called the third ventricle. All right, and then we have one here, and hopefully I don't take off the cerebellum, but there's a cavity right here called the fourth ventricle. So we have the two lateral ventricles, one, and then the other side is two. We have the third one here, which is the third ventricle, and then we have the fourth ventricle. Now, these ventricles, they're openings where what we call cerebral spinal fluid travels throughout the brain. All right, so cerebral spinal fluid is a very clear liquid that helps nourish, that helps provide protection and cushioning for the brain. So if you want to see that imaging, so we got this image, really great image of our, from the Crump Institute for Biological Imaging out in UCLA, I think, if I'm not mistaken. So they have an imaging that shows us the flow of that CSF, the cerebral spinal fluid. So it actually bathes the brain throughout. So it actually goes from the very center of that third ventricle, kind of spreads out to the lateral ventricles, then go to the fourth, and it actually spreads out to the spinal cord. So there is a canal that goes all the way down to the spinal cord. Okay? So, with that being said, it also gives weightlessness to the brain. All right? So that's why we don't feel that 3-pound weight in our heads, because it kind of flows very weightless up there. All right, excellent. So let's go back to our sagittal cut. So this is the cut that we are looking at. So when we make a cut, when we make that dissection, all right, that goes along the fissure, that means it's a sagittal cut. All right, it's kind of going from vertically. All right, so we have our ventricles. The next thing we see here is this white structure here. So let me see if I can zoom in a little bit. Right here, this white structure is called the corpus callosum. The corpus callosum is also a bundle of nerve fibers. So think of it as like our spinal cord. But you notice that it's kind of like a comma shaped that is found on this side of the brain, and it's also found on the other side of the brain, on the other cut. This is the bridge between the two hemispheres. This is how both the left and right hemisphere communicate with one another, through this corpus callosum. All right? It helps work both hemispheres together to get those motor senses running, those sensory-ish information running. All right? It handles all of that business between the left and right hemisphere so they can communicate properly and perform the functions that the brain is asking the body to do. All right? So we have the ventricles, we have the corpus callosum. All right, the next one. You notice this little structure right here? That's called the pineal body. So the pineal body is actually a gland that's found in our brain. It's one of the many glands that we have in our body. The pineal gland helps with what we call our circadian rhythm. So our circadian rhythm, so pretty much this area of this structure right here helps us with sleep. It's our internal clocks. It determines when we should be aware and awake and active and when it's time for us to go to sleep. So if you're wondering what the gland excretes to perform this internal rhythm of being awake and asleep, this cycle, it's called melatonin. So when more melatonin is introduced, excreted from this gland, it's time for us to go to bed. When there's little melatonin within being excreted, that means we are wide awake, all right? Now, obviously, you might ask them, why do adults need coffee? That's an adult thing. We just need it because it's just a nice little boost, all right? But for the most part, the pineal body excretes this melatonin or, stops that melatonin excretion to keep us awake or asleep, all right? So here we go. So that's our pineal body, all right? So the next structure that I want to talk about is right in front of the pineal body. All right, so this right here is called the thalamus, and then it kind of stretches out. There is no division here, but there's like little white linings that you can't see on screen. The hypothalamus is located here. All right, so the thalamus is the larger portion. So that's the region between the midbrain, all right, and the rest of the brain, okay? It receives all different kinds of sensory signals and relays them to the rest of the brain, to the cerebrum, all right, to the cortex here, the cerebral cortex, all right? So it acts as that relay for information. The hypothalamus is a small structure, super important, and it helps with hormone secretion, all right? It helps with any of hormones that need to be sent out to the rest of the body. It also helps regulate temperature. All right, so that is a very big, important thing for us. All right, as humans, all right, we have the ability to regulate our body temperature. So the hypothalamus helps with that function to keep us at a nice average 98.6 degrees Celsius. Not Celsius, Fahrenheit, excuse me. Fahrenheit. So 98.6 degrees Fahrenheit. All right, so if we were to get cold, the hypothalamus helps us, start shivering to warm us up. If we are too hot, we start sweating. So the hypothalamus regulates all that internal body temperature. Okay, so let's move on. So, the next thing we see here, so let's look at the cerebellum because it actually has a nice little design going on inside. So someone mentioned gray matter and white matter. All right, gray matter and white matter. So that's very easy to defer here. So you see this little pathway? So it kind of looks like a tree actually. In fact, the structure is called the arbor vitae, which is Latin for tree of life. So that's how the makeup of inside the cerebellum looks like. All right, all that white line that you see there, that is white matter. So that white matter is actually myelinated nerve fibers. So if we look at a nerve cell, we call this the axon. So that's actually what sends all that information to all the other nerve cells within the brain or throughout the spinal cord. All right, communicate throughout the nervous system. So the white is that sheet. So these little appendages of the nerve cell have little white sheaths to cause that communication to go very fast. The gray matter is the nerve cell bodies themselves. All right, so white matter is that extension of that nerve cell. The gray matter are all those little nerve cell bodies. All right, so let's look at one more structure. All right, and it's this structure right here. This is the superior colliculus. All right, so this portion helps coordinate movements of the eyes, the head, the neck, in response to stimuli. So I'll go a little more into detail about that. But underneath is called the inferior colliculus. So that deals with auditory information, so sound. So the best way for me to describe the superior inferior colliculus is now that the weather is warmer, you notice that there's a lot of buzzing insects going around, whether they're bees, mosquitoes, flies. So let's say, you're hanging out in your living room and all of a sudden you see a fly pass by. All right, your brain is going to register that stimulus. That stimulus is that buzzing fly going about your living room. The superior and inferior colliculus, depending on how close that fly is, you can hear it. It's going to relay all that information, gather all that information, and tell the brain and the rest of the body how to react to it. So that's why when you see a fly buzz by right in front of you, you start following it and trying to look where it's going. That is your colliculi working together, especially with the sound, trying to pinpoint where that fly is going. All right, so that's a nice little simple example of what those little structures do within the brain. Okay, so one of the really cool things here actually is we have a nice cross section of the optic nerve. All right, and if we put it all together, all right, so we have our diagram here of the sagittal cut. And those are all the parts right there. So we have the spinal cord, all right? We have the gyrus, so you can see the little bulges there, all right? The little linings from the sulci. I know someone mentioned the coloring of the sulci. Is that because of the preservative or they're naturally that way? It's because of the way that the organ is preserved in these chemicals as preservative to make sure it doesn't decompose. Sometimes, because of the way that it's preserved, it kind of decolorizes, and it just naturally, because of the interaction of that chemical, kind of gives us that nice little dye view. So we have the hypothalamus and thalamus. We have the ventricles. All right, 1, 2, 3, and 4. So 1, 2, 3, and 4. I should do it correctly. All right, we have the inferior and superior colliculus. So let me get it on the screen there. We have the arbor vitae, which is inside the cerebellum. We have the medulla, the cross-section of the medulla, cross-section of the pons. We have the spinal cord back here. All right, and that's pretty much the sagittal cuts. So we pretty much went through a lot of anatomy through this dissection. We can delve into deeper. There's a whole lot of information that we can look at in terms of the brain. These are just a good list of some of those parts that we can see through this type of specimen. Obviously, I didn't go through all the nerves. There are 12 nerves that are found along the length of the brain that are called the craniofacial nerves. All right, so there's 12 of them. Okay, and you can see some of those little nubs, but however, we can't see them without the usage of a microscope. I also kind of previewed a little bit of the meninges. So the meninges right here are the extra protective layers that envelop around the brain, the spinal cord. All right, this is where there are 3 layers. So if we were to, so these types of specimens do not have the dura or the meninges kind of wrapped around it. So if I had my brain here, my hand is kind of like the covering. All right, that protects the brain from the outside environment. All right, so you may have heard of meningitis as an illness. So that is the infection of these 3 layers. So the dura mater is the toughest of the 3, it's Latin for tough mother, and that's the outermost layer that protects the brain from everything else. Within the 2 layers are transparent, so that's the arachnoid and pia mater, and those are transparent, and that's where the CSF, the cerebral spinal fluid, flows through to replenish, to give nutrition to the brain, so on and so forth. Most of the major blood vessels are found within these 2 layers. All right, so that brings us towards the end of our dissection. I spoke a lot. Hopefully, Ms. Caroline and Ms. Rosa were answering as many questions as they can. I know there was a lot of questions. All right, so let me go through and see if I can answer some of those right now. Before I go through all those questions, all right, as a reminder, thank you all for joining us for our live stream. This is your first time. Hopefully you can come back for more. If you are a regular for our live from program live streams, welcome back. I hope, just want to give you a nice little excitement, hype you up. For the month of June, we got some really awesome programs for the live from surgery that coming up. We actually have some of the surgeons from our live from surgery program actually coming in, joining us for our live stream, giving us some information about their careers, showing us some really cool things that they do on their job. So if you are interested in checking those out and get inspired to possibly be going into surgery or becoming a nurse, so on and so forth, June is definitely the month to check those out. Also, if you're interested in astronomy, we have our planetarium live streams every Thursday at 1 p.m. We have Mr. Kengo, Ms. Caroline doing LC Junior, so those are for our younger audience members. Those happen on Mondays at 11:30. We also have trivia that happen on Thursday nights for adults. We have family trivia on Fridays in the morning at 11:30, and we have our animal updates. If you're curious about all of our animals at Liberty Science Center while we are closed for the time being and working from home and learning from home, definitely check those out tomorrow or Wednesdays, I should say, at 1 p.m. We have other stuff too for you to check out at lse.org for LSE in the House. Lots of experiments, other videos from the past. Definitely check those out. Hashtag us Liberty Science Center. Show us that you are interacting with us. We definitely want to see all you scientists out there performing those experiments. Once again, a special shout out to our members and our major supporters for keeping us going, providing you all this content every single day. It's a lot of work to put Monday through Friday programming for you all. All right, so if it wasn't for our members, our supporters, those that are going through lsd.org/donate to, help us out, we wouldn't be having these live streams right now. All right, so let's see. Let's see some of those questions. So I'll do a couple of questions for 5 minutes. Is it true, this is from Patricia, is it true does the left side control the right side of the body and the right side of the brain control the left side of the body? That is a very good question. That is absolutely true. All right, so that goes back to the optic chiasm. All right, so you notice that I mentioned that the optic nerve kind of crisscrosses and then heads into the brain. So it's the same idea with the right side of my body is controlled by the left side of the brain. All right, the right side of the brain controls the left side of the body. That's why the corpus callosum is super important to bridge those communications together. All right, if there is some sort of damage to the corpus callosum or some type of communication pathway there, there could be a detriment to either side of the body. One of the biggest conditions that is known to cause some of that type of damage or aphasia is when an individual has a stroke. All right, depending on the location of the stroke, if it's a right-sided stroke, sometimes the left side of the body may be limp or there's not much function going on there. Same thing if it's a left-sided stroke, it would be for the right side. All right, what do sheep eat? Very good question. They eat grass, depending if they're at a farm. They, they graze through the grass. They were given hay. They're plant eaters. Very good question. All right, do brains control sweating from Ronnie, and do sheep dream? All right, so let me tackle the brains control sweating. So that goes back to the hypothalamus. So your brain is a computer, all right? It's the supercomputer for your body. Doesn't necessarily mean that they directly control sweating. What it does is that this supercomputer, all right, sends out signals to perform those functions, to jumpstart those mechanisms to perform a function. So right now it's super hot and your body senses that it's rising in heat temperature, all right, its body heat is rising and you need to cool yourself down. Your hypothalamus figures that out, gets that information from the rest of your body that it's heating up, sends out a signal back to start all those sweat glands to start profusing that sweat to start controlling and cooling and bringing your body temperature down. So your brain handles all of those functions. There's a lot of mechanism, a lot of feedback, a lot of communication that goes on from your brain to the rest of your body, to the rest of your body back to the brain. All right, 24/7 to ensure that everything is running smoothly as an individual. All right, so let's see. Any other questions? How old can a brain get? Very good question. So it gets as old as you get old. So all the organs in your body, including your brain, right now I'm in my 30s. All my organs are in their 30s. All right. If you know someone who's 50, all their organs are 50. So, it's relative to the age. Okay. So, there is no stoppage as to where, does the brain stop aging? Does your organ stop aging? It all depends on how long you live your life. Now, there are conditions that, maybe as you get older, you might lose some sensory stimuli, sensory processes here and there, but that comes with age. Same thing with vision. As you get older, sometimes your vision might weaken, okay? So those are just as a result of becoming older. But there is no stoppage as to, does the brain stop aging at a certain point? It just ages along as you age. All right, so is the sheep brain like the human brain? Absolutely. So that's a very excellent question. That's a very honest question. So we wouldn't be doing this dissection if we were not learning from it. All right. If we wouldn't be doing this dissection if we weren't going to be learning anything about our own anatomy. All right. So that goes back to comparative anatomy. So there's a lot of structures that happen within the sheep brain, a lot of structures that we can examine that are extremely similar in structure within the human brain. Now, there will be differences. Like I mentioned, the human brain has a bigger frontal lobe than a sheep. Sheep has stronger, more pronounced olfactory bulbs, optic tracts, all right? And that's because it's all based on the adaptations that they need. We don't have those as pronounced. If we had those pronounced, we'd have a long snout to pick up all those smells, right? But if it wasn't for all those similarities, we would not be doing these dissections whatsoever. Same thing with the sheep heart, same thing with cow eyes. we would not be doing these dissections if we know that for sure that we would not be learning anything from it. All right, so let's see. Other questions. Why is the sheep brain yellow? Excellent question. That's because of the preservative. Because of the preservative that is used by the company to make sure that the brain doesn't decompose, and also I have a light, so depending on my brightness level and everything, it looks very yellow in color. But brains are very pink in color. All right, they're not like super bright pink, but they are a pinkish tinge to it. So excellent question. I'll take probably 2 more, and I will end it there. So as a reminder, if you are a teacher or if you had homeschool students that didn't have the ability to check these out, definitely check out lsc.org. Go to our LSC in the house page. We will definitely have the video up after the broadcast. So give it about, later on in the day, it should be up. My colleague Jeremy will handle all of that for you all. So you can definitely go back, check it out tomorrow, check it out next week, the next month. You can definitely check it out on our page. What makes you faint after you get hit in the head? So when it comes to fainting, a lot of the act of fainting is the result of not enough blood circulation going through your brain. There is a lot of brain circulation going on inside your head right now. Your brain may be small relative to the rest of your body, but it uses up 20% of the body energy to make it work and function correctly. So that means it needs a lot of blood flow to go into your brain. All right, so that means that if we were to fill up soda cans of the amount of blood that circulates in our brain, we can actually fill up 3 soda cans of blood, and that's how much blood is circulating constantly throughout our heads. All right, it just needs all that energy, needs all that nutrition, that oxygen. So a lack of blood circulating through the brain, you would end up getting lightheaded, and eventually you can pass out. And that's just a reactionary mechanism to ensure that your brain doesn't overheat, and kind of conserves and waits until the blood kind of comes back into circulation to provide that energy, all that nutrition again. Yeah, I can definitely turn off the light. So there you go. So it's still yellow in color. So there you go. All right, and I'll do one more question. And then we'll end it there. How does the brain know what to do and when? Excellent question. So that goes back to the idea that your brain is a supercomputer. All right. Right now, as you're sitting watching this live stream, you're already gaining information. You're looking at your screen. You're getting all that information. The information is being sent to the brain. Brain's being processing it, interpreting it, sending it back. As you're sitting, you probably kind of feel like the chair, or you're kind of rubbing your hands together. You probably have all this peripheral vision. You have so much information going on in your environment. All right, and you have all these different sensors and nerves all throughout your body. All right, you may not notice it, but these nerve cells, these sensors, they're going, super quick, even at a millisecond rate. All right, all this information is being sent to the brain. Brain's processing it at super, super fast speeds, and then sending that information out. All right, so you can see I'm like waving all my arms and all that. That's my brain telling me to wave all my arms around like that. All right, so all this information, it just comes through. So when does the brain know what to do? At what point? That's a little bit more specific. So let's say, you're watching a scary movie and something comes out on screen and it scares you. That's your brain giving you, at that moment, it says, all right, something scary just showed up. We got to do something about it. We got to run away, or, be on the alert that something else might be wrong going on in the environment. At those specific moments, that's when the brain kind of gives that extra oomph in terms of communication. All right, so I did a lot of talking. Hopefully you learned a lot. Hopefully I showed you as much of the structures as I could. All right, I know that this brain was a nice specimen to look at all those structures. And hopefully it inspired you to become scientists, maybe go into the field of neurology, learn more about the brain, because there's still a lot of stuff that we don't understand about our own brains that a lot of studies are still going out for. Hopefully that inspires you all, and hope to see you next week for our next Live from Surgery program on Tuesday at 1 p.m. My name is Alejandro, one of the STEM Educators LSC. Enjoy the rest of your day. Thank you. You're all awesome.